Mhm. So today we are gonna study about receptors. Okay. So as I have covered three topics before: the transporter, ion channel, and enzymes. The receptors will see. Okay. How the drug is going to get attached, what is the process that happen, and how we get the action, what are the time it takes, why it takes macromolecularly, where it attached to protein or to the surface, or what type of channels they have, what different type of receptors there are, so that the drug can go and start the action. That can be agonist action, antagonist action, to stop something, to increase the activity, or to stop the activity, for a while. So these are the factors that these are the results we need, and the factors that will affect, and the what is the major role of receptors in this. Okay. So receptors are, they are the protein structure present on the mammalian cell or within the cell. Now these receptors are either present on the surface of our cell membrane, like G protein, or sometimes they are present intracellularly inside the cell, lipids, or in the nucleus, where they have their own channels and that they do the actions. Receptors are sensing elements in the system of chemical communication that coordinates the function and function of all different cells in the body. The chemical messenger being various, they can be hormones, they can be neuroreceptors or other mediators. It depends on all the things will depends on the receptors. Okay. Many many therapeutical useful drugs act either as agonist or antagonist on receptors for no endogenous mediators. So they either will increase the activity while connecting with the receptors, they will or they will work as an antagonist to stop that activity if there is overdue of something. If there is, let's take a example, if there is a cancerous tissues, so now the drugs will work as antagonists to stop that, to stop the malignancy of it. Now in Parkinson, as a dopamine, it will increase the activity. It will take the dopamine to that particular place, and that will increase the activity. In thyroid, we use some drugs to stop the activity. So according to our need, or according to the need of the region, the drug will work as agonist, antagonist, and there are two more other, irreversible and partial, that I'll explain to them. So the types are: receptor elicit many different types of cellular effect based on the molecular structure and the nature of transduction mechanism. Four types of can be distinguished. So these are the four types of receptors we have: neurotransmitters, synaptic, anything related to our nerves, anything related to our brain, that would be gone under this synaptic. They are very fast and give you the operating time of milliseconds. Okay. The other is catecholamines. It will also give you in seconds. This is again uh, to our related to our body, to our brain. The thyroid and steroid hormones, this will take few hours or days to do the activity and get converted. Now this time that hours and days has been given is, the drug will come to contact of the cell, and it will take at least an hour or 45 minutes or any minutes or even to give the result. Some chronic disease or some disease are like organ oriented, depending on the administration, depending on the route and everything. After that route has been decided, once the drug go to that cell, how much time it will take once it come in contact with the receptors. So the if we are going to see any drugs related to thyroid, steroid hormones, we can expect the drug affinity, means the pace at which the drug will work would be hours to days. Anything related to the brain, the acetylcholine are present in the ion channel. In the receptors, the acetylcholine would be present, the synaptic molecule, and the result would be easily taken place. Okay. The fourth is peptide. It will take longer time. This is basically for the lipids or the protein that are present over there. So one more major thing you have to remember is protein plays a very important role when it comes to receptors. There are major majority of drugs that will get attached to protein. So you have to, when you go through the medical documents or something, we have to be very much known what are the protein percentage they have in their body. They take the drug that we decide, or anyone, the doctor decide, take account of the protein level as well. So they will increase, or they will ask them, they will supplement some kind of protein as well. When the drug, which will get affinited with the receptors of protein, they will require certain level of protein if they don't have. So accordingly, the supplementation will be done. So whenever it comes to receptors, whenever it comes to drug, it's just not the route that we choose. It also the macromolecules that are present and are being affected as we are going to treat the patients. So at many level, the affection would be there. The gut won't be perfect. They might not be in conscious level. So that's the reason we take the blood reports, we take the lipid reports, we take all macromolecules reports just to administrate what type of drug is being given. So to perform one function, we might have different drugs. So in future, we will be learning, we will be going through those things as well, but have a clear mind about it, why this drug was used. Sometimes what happened for a fever or very small thing, we can use normal paracetamol, but as the certain quantity or the certain affinity of the drug won't be present for the patient to handle it, we use the combination of it. We use low mg or we use more mg of it as we want the result according the pace of it. Okay. So there are four type of it. First is ligate gate ion channel. This is the normal. The receptors are present at the surface. Okay. The first is ligate gate. The time scale is milliseconds. Example is nicotine and acetylcholine receptor. These are, as I told you, milliseconds. Whenever it will come, millisecond is synaptic transmission. So whenever any drug will come in CNS, so you should know or you should be aware that it's going to perform in millisecond, and this type of ligate gate ion channels are being used, and which drug will be coming under ligate gate ion channels, that we will learn again in CNS. But right now just have a overview of it, because this is the basic of drug administration. So this won't be coming in further lectures. Now we'll just learn about the what are the drugs are being used and what they do when they go there. Okay. So in ligate, what happened? These are the membrane protein. Membrane protein means they are present on the membrane layers. The protein are present on the surface of it with the similar structures of other ion channel and incorporate a ligate binding receptors site, usually in extracellular domain. Extracellular means out of the cell, on the surface of it. On this receptors, fast neurotransmitter act. Means acetylcholine would be present over there. The drug will come and it will get attached with acetylcholine and do the work. Example include, these are the examples: GABA receptors, NMDA, glutamate receptors. These are the drugs that we use. Okay. It's okay that if you don't remember this right now, once you go in CNS, once we'll go through those systems, you will get a better idea why the GABA, what they mean by GABA receptors or glutamate receptors. Okay. So the first is ligate gate ion channel. That is for acetylcholine neurotransmitter act. In this, what happened? What is the structure of it? Or what is first is this happens. See, they have this four structure inbuilt in them. The fragments of the subunits are alpha, beta, gamma, and alpha. Okay, so now what happened? This is the structure, the main diameter. It's like this. This acetylcholine is the receptor. So once the signal will come, it will go to this acetylcholine and they will bind the domain and do the action. Okay. The first is it assembled from four different type of subunits. These subunits are inserted into the membrane outside on the surface of the cell. The oligometric structure possesses two acetylcholine binding sites, each lying at the intersurface between one of the two subunits and its neighbor. So now two acetylcholine bindings are there in one ligate gate ion channel receptors. Both must bind acetylcholine molecule in order for the receptor to be activated. Okay, so whenever the drug will come, it will come in contact with this to acetylcholine, and then and only then this gate will get activated for the function whatever is being required. Okay, so whenever someone asks or is asked in the exam that explain ligand gate ion channel, so you just have to write a little definition of receptors, the types of receptors you will write, what are the four types, and then you just write the link, you'll just explain the ligand gate channel ionotropic with this diagram, and then you will draw this diagram. Any one of this you can draw. You can draw the first domain, that is that, or you can just uh, make a strand of four subunits that are present in linkage. Okay. Now the second is G protein coupled receptors, GPCR metatropic receptors. In this, what happened now, as the name suggests, the receptors are present in the protein. The drug will get bind with the protein. Okay, it might be on the surface or it might be inside the cell, depending. Now, these membrane receptors that are coupled to intracellular effector system via G protein. So they are on the surface as well, but they are connected to the intracellular work. Means inside the cell, they are connected through the protein. So when the drug will come, well, just imagine that one particular cell, epithelial cell or squamous cell, they have to work on that. And it's the drug is protein binding drug. So what will happen? The drug will come and get attached to this point, G protein, the receptors, then G protein, and then it will go to the messenger. Then inside the cell, the protein perforation, other calcium will be released, cellular effect will be there. Okay, so this also takes a few seconds as a response to happen. The ion will come, it will get accepted by the receptor, G protein receptors, and then it will go inside the cell. Okay, they are the membrane receptors, they are coupled with intracellular. They constitute the large family and include receptors of many hormones and slow transmitters. Example, the muscarine, acetylcholine receptor, adrenoreceptor, chemokine receptor. Subtypes are also present and all are the basic structure. So you should remember that when it comes to G couple, then what you'll do, you'll make a normal cell structure, you'll put a ion channel, and then you'll draw a receptors that is getting attached with the G protein. Okay, the drug will come, get attached to the G protein, then and only then it will come into contact with the inner cellular level and do the function. Okay, then this thing cellular effect will come, calcium effect will come, that those are the materials which are present in the cell. They will get activated as per the requirement. The second is G protein couple. Now the third is kinase link related receptors. Okay, these are also protein mediator. Okay, it will come and it will directly go inside the uh, cell, and the protein, first what will happen, the protein phosphorylation will happen, gene transfusion, protein synthesis, and then and only then cellular effect will come. So when it comes to protein, so the drug will come in contact with receptors and then it will get bind with the protein. So the process of protein bindation, gene transmission, protein synthesis will take place, and then we will get the effect of drugs. Now for this, it will take few hours as well. So kinase linked receptors, it will, if the drug is affinited to kinase linked receptors, it will take few hours to give the effect. So whenever we need effect till night or something, it will be administered in morning or in afternoon, in noon time. And then you can expect after 3 hours, 1 hour, whatever the time it will be required, but it would be in hours. And then the effect will come and they will feel better. So what happened in this is a large heterogeneous group of membrane receptors responding mainly to protein mediators. Okay, they comprise of extracellular ligand binding domain into the intracellular domain by the single transmembrane helix. Now what happened? This is the single. Now gene transmission, what, just one receptor will be there. that will be connecting the extracellular and the intracellular. The drug would be coming from the blood, that is extracellular. It will connect to the receptor, it will go into the cell, that is intracellular. There it will take transcription of gene and protein synthesis will happen. Drug will make some metabolites, some other complexes, and then cellular effect will be coming. So before the cellular effect, there is very much important that the presence of protein should be there. In many cases, intracellular domain is enzyme in nature with the protein kinase or glutamine cyclase activity. Example is type 3 receptors include all those insulin and for various cytokinase. Okay, sometimes what happen, insulin resistant, the cell become insulin resistant. So what happen, all the sugar content or the all the glucose are running in the bloodstream. They cannot enter the cell because they are insulin resistant. So what will happen? The drug will come and take that glucose through the insulin and get all those glucose which is running in the blood, which shows the high glucose level in sugar patient. It will go inside and it will shift all the glucose which is running in the bloodstream into the cell. Okay, so that is the example of it. So in kinase linked related receptors, what will happen? You just have to remember in this, it is protein mediator. So protein is needed for the drug to travel to intracellular and give the cellular effect. Okay, and it will take hours. The first will take few seconds. Second one will also G protein coupled receptors will also take few seconds, and then the kinase link receptors, it will take hours to give the effect. The fourth most is nuclear receptors. These are the receptor regulated by the gene transmitter. When we say gene transmitter, it comes to chromosome level. What particular gene would be present, should be present in that cell to get that drug effect. Okay, then it will go and have the protein synthesis, and then we will have the cellular effect. The term nuclear receptor is something of a micromer because some are actually located in the cytosol or and migrate into the nuclear compartment where when a ligand is present. They include the receptor for steroid hormones, thyroid hormones, and other agents such as retinoid or vitamin D. As suggested as explained before, this first one, thyroid hormone. So now you know how for the thyroid hormone, what kind of ion channel will be there. So you can expect a question like this. Okay, for thyroid hormone, give an example of a receptor. What kind of ion channel will be used for the thyroid hormone? So then you have to give a definition of receptor. You will write the 4 types of receptors, and then you will explain this one, fourth one, nuclear receptors. Okay, so just remember the examples and remember how long it is taking to give the effect. And then you just have to, you can explain with this or you can just make a diagram. It will be a 5 mark of questions. Okay. Nucleus, then gene transmitter, and then protein synthesis will be there and cellular effect will be coming. So everything is happening in the nucleus. That is why the name is nuclear receptors. So the drug is coming, it is going to the nucleus, the receptor is present in the nucleus. There will be a gene transmitter which is present, that is very much important for the drug to come in contact with protein and give the cellular effect. And this all process will take few hours or days as well to give the effect. So as we say, we give vitamin D injections weekly, monthly, or sometimes monthly or 6 months one B complex. Those are the injections we give because we know every 1 month it will take certain time to complete the action. So we do not give every day or weekly when we know it's going to take days to give the effect. So now I'll just explain you what is the concept of receptors, what is happening. It will go there and how it's going to function, what are the levels. So this is the history of the receptor concept. I'll just read it out. What are the who just explained it for the first time and what is the definition. So to give a definition for receptors, which is written this in red, you have to put that as a definition. Okay. Then you'll just write about the types and everything, and then you'll explain the type. The concept of drug acting on receptors generally is credited to John Langley while studying the antagonist effect of atropine against the pilocarpine includes salivation. The word receptors was introduced in 1909 by Paul Andlich, postulated that a drug could have a therapeutic effect only if it has a right sort of affinity. Now, what do you mean by right sort of affinity? Means just imagine that there's a room and there's a particular three different type of gates are there. Okay, if everyone which is present outside the door are trying to get into it, but if the drug have a particular key to that door, only those drug will get enter in that room and the effect will come. So the drug should have that affinity to get into the door. So in pharmacology, what happened? We need to know through which door we have to have the action. We need the action from which door or which cell at what level of at what time. If we need the action in few seconds, in hours, or in days, or in the other hand, when we know that this drug will take this much of time, so according to that, the dosage would be decided. As we say, this medicine should be given once a day. You can take thrice a day as well. When we know the affinity will be more, that the drug will be absorbed and give the action easily, and the dosage is less, so we need more of it. So we'll give it frequently. We'll just ask the patient to take it twice a day. Okay. The drug receptor, the definition is the receptor are specialized target macromolecule present on the cell surface and intracellular receptors. They are present on the surface as well as in the intracellular, maybe in the normal cell, in the nucleus, or in the cell G protein, as an example of G protein, or on the surface, bind the drug and initiate the event leading to alteration in biochemical and biophysical activity of the cell and consequently the function of a drug organ. Okay, the drug receptors. From a numerical standpoint, protein from the most important class of drug receptors. Example include the receptors for hormone, growth factor, transcription factors, neurotransmitter, the enzyme, dihydrofolate, reductase, acetylcholinease, cyclic nucleotide, phosphodiesterase. Now this is very important, drug receptor interaction. What happens when the drug comes in contact with the receptors? Then what happened when this both go in contact with the cell and how the action is being produced. Okay, so the binding of the drug of receptors can involve all known type of interaction. Some binding is going to happen between the drug and the receptor, then and only then they will be able to uh, come in contact with cell. Okay, so what are the bonds that are happening between the drug and the receptors? The first is normal ionic, second is hydrogen bond, covalent bond, van der Waals or hydrophilic. Hydrophilic H2O, van der Waal is a molecular calcium or potassium. In covalent, carbon bond. Most interaction between drugs and their receptors involve bond of multiple. If the drugs of drug action is frequently but not necessarily prolonged. Non-covalent interaction, high affinity will be there. Okay, so the just go through this K1, K2, K3. I'll explain to you. The drug plus receptors. The magnitude of the response is proportional to the number of drug receptor complex. Means what happened? The drug. Hmm. So what will happen? So the drug will come in contact with receptors. Okay, then this drug and receptors will, the rate will define that how much time the drug is going to take to do the action. If at the first stage only the drug is not coming in contact with receptors for some reason, okay, the action is going to get affected. So that is the first stage, where the K1, K2, K3 are the rate of constant. The rate at which the drug molecule combines with the receptor site is K1, at what rate the drug is going to get attached with receptors. Similarly, the rate at which the drug receptors, now the drug has come in contact with receptors. Now the stage 2 will come, where this drug receptor complex will dissociate, is given by K2. Dissociate means it will get detached. Now the drug has come in contact with receptors, and receptors has given the allowment that it can go in intracellular or it can go inside. Okay, so that is the second stage. The third stage is the rate at which the response is generated following the drug receptor interaction is given by the rate K3. The response is this cellular effect. So the here K1 is when it came to the receptor, that is 1. Now the from receptor it is going to gene transmission doing protein synthesis, that is 2. Once it is completed, it will give you the cellular effect, that is stage 3. The constant are used to define some basic concept related to the drug receptor interaction. In general, the drug receptor interaction is characterized by first by binding of drug to receptors. That's clear. The first is the interaction of it. The second is generation of response in biological system. Now the drug has come in contact with receptors. Now drug is allowed to do the effect. Now it will change its affinity, it will change its content. The biological system, there will be a change in biological system. The first function is governed by the chemical property of affinity, means how fast the drug can come in contact with receptors. That is affinity. That is ruled by the some chemical forces that cause the drug to associate it reversibly with the receptors. Now, the affinity, what is the definition of affinity? Is it describes the ability of the drug to form and subsequently maintain a complex with the receptor site. Affinity means what? The ability of the drug to come in contact with receptor and to stay in the contact with the receptor as much as is needed. What if it comes and it doesn't stay? It will go back to the bloodstream and it won't act. That is called drug resistance. Okay. The second is intrinsic activity. Now the drug has come in contact with the receptors. Now we have a complex, drug receptor complex. How fast they are going to function together to get us to the cellular function. That is intrinsic activity. It plays very important role. The more the intrinsic activity, the fast the drug will give the effect. The fast we will be having the result. Okay. It describes the ability of the drug to evoke the pharmacological response on combining with the receptors. Okay, the potency and efficiency. Potency means a drug is said to be potent when it possesses high intrinsic activity at low unit of weight dosage. Means just imagine giving any medicine, or if I'm giving 10 mg, okay, I just want a result that the fever should come down. The person is having a fever of 101 or 103. If I'm choosing, let's just say A, B, and C, there are three drugs that I have. The A drug at the content of 10 mg, B drug at the content of 200 mg, and the C drug with the content of 600 mg. They all are going to give me the same effect in 10 minutes. So how you're going to judge the affinity of it? So the 10 mg with the least dosage, they are going to perform the intrinsic activity very fast and very accurately, so that the action will come. In response to other, the 100 mg and 650 mg, what will happen? They will go, the root will take some absorption, some level of a dose will get wasted over there. Just say it will get wasted in gut, it will get through the first metabolism in liver. Okay, then it is getting wasted in receptors, it won't have a affinity to that level. and then it will go to the nucleus and work that. So it's going to take longer time, as we need more amount of dose, because we know as the route of administration needs some amount, there would be some amount of wastage of dosage is going to happen over there. So the amount of the dose is very much important. If the amount of dose is least and you're getting the action, whatever you need in short period of time, that will signify the drug ability. The efficiencies, it refer to the maximal or the peak response produced by a drug and is an important determinant in the drug selection process. Okay, how
Yeah.
quick response the drug will be giving to us, that is the efficiency. As a normal definition, we know that at what efficiency you
Yeah.
can work. Okay, so these are the different terms that are being used for drug and receptors. The first is agonist. What happen in this? The drug will go there and get attached to receptors and it will give the function. It will get the activity done. That is agonist. What was the purpose of the drug? He is doing that. That is agonist. What is antagonist? The drug will go and get attached to the receptor and it will stop the function. Just imagine in cancer patient, the cell are growing in multiple rate. Okay, so now the drug will go there, attach to the receptor, and stop that multiplication. Okay, it will stop the process. So that the name, the function of that drug is as an antagonist because it's going to stop the activity over there. The third is partial agonist. So what happened? It will go there and it will stop to some extent. It will take few hours to respond, and it might not even decrease. It won't stop it. It will reduce it. If we want to go as a partial agonist, so what will happen? The activity will be increased, but in a partial manner. If we go as an antagonist, partial antagonist, what will happen? We want to block the activity, but we don't want to stop it. We just want it to get reduced. Okay, we just want the temperature to come down. We don't want the temperature to go in minus. We want it to maintain. So to some extent, partial antagonist, we're going to use such a drug that will work as a partial antagonist over there. In the fourth is inverse agonist. In this, what happened? In the drug that cause an effect opposite to that of the agonist. In contrast, means no matter what you give, whatever the function there is, it will go reverse it. Okay, so if you want to raise a temperature, so you'll give us some drug and that will reduce the temperature. So the drug will go and function antagonistically. In this drug, competitive antagonists that simplify block the agonist but has no inherent action. The drug won't be having any action. It will just go to the receptor and whatever. If the receptor is working as an agonist, this drug will go and stop it. If the drug is working, if the receptor is working as antagonist in that cell, the drug will go and activate that activity. So there won't be a purpose to it, there won't be a rate to it. It will just go and attach to the receptor and it will reverse the function. That is inverse agonist. The drug doesn't know that I have to increase to this level. There won't be such thing. Whatever the receptor function is, it will get reversed when it will come once it will come in contact with drug. That is inverse agonist. So it was a big topic. I hope it was understood to some level. Once you'll read it out, you'll have a good idea about it. Anyone have any doubt regarding anything?
No, madam. Thank you.
It was clear. It was easy for you
Yeah.
because okay. So we have completed this, the administration of the drug, the action of the drug. Okay. Then we'll have the different topic. Tomorrow we'll have a different topic. Okay. If just go through this on Thursday. I think so. We just have three lectures this week. Mainly, I'll just take one more on Thursday. I'll take a test of this because this is an important topic. So I want you all to go through it. On Thursday, we'll be having whatever I'm going to make you study in this week. We'll have a test on Thursday. Okay.
Okay, ma'am.
So I'll just share this PPT if I haven't. So I'll share this today and just go through it within 2 days. Okay, if anyone have any doubt regarding this, you can ask me tomorrow. You can ask me to explain it again so that I can clear some doubt if there's any. Okay.
Okay.
Okay.